Hey, science geeks. Welcome to Chapter 14. This is Chapter 14, Principles of Disease and Epidemiology. So we'll be looking at transmission of disease, a lot of terminology related to disease and the tracking and tracing of disease. And then what is epidemiology and how is that related to disease and how it spreads. This will be a three-part series, three-part lecture series on this topic. So let's get started. So some terminology. Pathology, the study of disease. These terms, of course, are related to disease itself, obviously, pathology being the study of disease, but also the spread of the disease. So we'll look at epidemiological terms as well. Like etiology, the study of the cause of a disease. An etiological agent, for example, what is the causative agent to this disease? Pathogenesis means development of a disease, and if you split that word into two parts, genesis meaning the beginning of, and the prefix pathos comes from disease or something bad that happens to the body. So pathogenesis, the beginning of a disease. Infection would be colonization by pathogens. And disease, a very general term for this, an abnormal state in which the body is not functioning normally. So we could use that for a wide variety of things, like even could say if you had a headache, I guess that's the body functioning a bit abnormally. And so could we classify a headache as a disease? Well, according to this definition, you could. Although if you have a headache, I don't suggest you go around telling people you have a disease. That would be, I think, a bit of an exaggeration, but maybe you want sympathy. I don't know. Give it a try, see how it works. Let me know. So let's talk about the normal microbiota that we have, and then some terms related to that and some concepts. So we have about 10 trillion cells. The number varies greatly, but somewhere in that ballpark we have a lot. Yet we contain about 10 times or 100 trillion bacterial cells that are living on us and in us. We're normally free of all microbes in utero. And lactobacilli are usually the first that we contact. If we are delivered vaginally, we'll run into the lactobacilli that are present in the female vagina. But before that, other than that, there's nothing. We're in a sterile environment in utero. And then we are introduced to those when we come into this world. So we breathe in these microbes, we ingest them, they contact our skin, the surface of our eyes, and so on. And so then we become colonized by all of these microbes, many of which are bacteria, but there are some yeast as well that are growing on us too. So those are our normal microbiota. Now, there are times whenever the microbiota they appear, but then they go away, and those are called transient microbiota. They may be present for days, weeks, or months, and then they disappear because of some changes that occur, maybe in the environment or around you, the external environment, or maybe your internal environment changes as well, hormonal fluctuations and so forth. That leads to the production of different compounds in the body. Those compounds, many of which, some of them can be utilized by bacteria, and certain species of them will then become prolific, but then after a while hormone production decreases, the particular chemical compound is not abundant, and so those bacteria will disappear. Or any microbe, doesn't have to be bacteria, but generally it is. Normal microbiota permanently colonize us. Under normal conditions, they're not going to cause disease. However, if they get the opportunity to do so, they can, and we call that opportunistic infection, where in their normal environment, under normal circumstances, they're not causing any problems whatsoever. But in some cases, when they're out of their environment, things change, then an opportunistic infection can form. And symbiosis is a relationship between normal microbiota and the host. And so symbiosis can be seen in three different ways, which we'll be looking at. So symbiosis is just living together, organisms living together. They're in a symbiotic relationship in some way. Commensalism, one organism is benefited and the other is unaffected. And so most of our normal microbiota are commensals, meaning they benefit and we are neither harmed nor helped, at least directly. Now, there is something called microbial antagonism, in which the normal microbiota prevent the growth of pathogenic organisms on us or in us. So just them being there kind of helps us, but they're not giving us anything directly. That would be an indirect benefit. Their presence is all that's necessary there. They're not producing anything. They're not giving us anything in return for us giving them something. So that's commensal. We are neither helped nor harmed, but they are definitely benefited. Mutualism, everybody wins. It's a both organisms benefit. So, for example, E. coli, they produce vitamin K and some B vitamins, which we absorb in our large intestine, and then they get nutrients from us. So they are producing something that we directly obtain and benefit from, and then we provide them something that they directly benefit from as well. So this is mutualism. This is a two-way win-win situation. Parasitism, one organism is benefited at the expense of another, and we call them pathogens now. So the host is harmed, we are the host, we are harmed, and the microbe is benefited in some way. And so they're going to cause illness, they're going to release toxins, they're going to harm us in some way. And that's a pathogenic relationship or a parasitic relationship in which they are deriving something from us at our expense. Some normal microbiota, as I mentioned just a couple minutes ago, they're opportunistic pathogens. They don't cause disease in the normal environment, but in another environment they can. So E. coli living in our large intestine, everybody's happy, it's all good. But they get out of the large intestine and they get into our urinary tract and they cause UTIs as a result of that. So that's an opportunistic infection. E. coli has been given the opportunity to infect, and that's why they're called opportunistic infections. And E. coli is a culprit there. Another one would be you get a UTI can be caused by Staph aureus. And we do have some Staph aureus living on us as normal microbiota, not much. We have another Staph epidermidis, which is a cousin of Staph aureus. But we do have some Staph aureus living on us, and if they're on our skin, fine. If they get in our urinary tract, they cause UTIs, so they create issues there. Another opportunistic infection. So where are a normal microbiota? In this picture here you can see the large intestine obviously is loaded with billions and billions of bacteria. E. coli, of course, being the prominent one, but there are many other species that are living in there as well. All over our skin, we have bacteria all over our skin, in our sweat glands, and in our hair follicles. Well, maybe not that deep, but in the sebaceous glands, which are attached to hair follicles. All through our mouths, our nasopharynx region, on the surface of our eyes, on the conjunctiva, in our urinary and genital systems, the lower urethra, and both sexes in the vagina and females. And so these are the areas where normal microbiota are living and residing. You'll notice that the small intestine is not listed there. Stomach, of course, isn't because its pH is so low. Small intestine, 21 feet, you'd think, boy, that'd be a perfect environment for bacteria to live. You got all that space and all those nutrients. Well, small intestine is moving food through it pretty rapidly through peristalsis, and so it's continually pushing the food through. Whereas when it gets to the large intestine, things really slow down. There's a lot of water absorption and nutrient absorption in the large intestine, and so things open up, slow down, and now bacteria have the opportunity to multiply and divide and live and reside in our large intestine. Some factors that affect the growth of normal microbiota are temperature, pH, oxygen and carbon dioxide availability, salinity, sunlight, age, diet, health, stress, climate, hygiene, occupation, lifestyle. So the point of that is, yes, I read them off. You could read that yourself. But the point is, of course, there are many things that affect the normal microbiota that are living in us and on us. So where we live, the job we have, who we're around, all those things are affecting our normal microbiota. So, for example, I'm a teacher and I'm around hundreds of kids every day, and I'm teaching them and instructing them, and I'm in an environment where microbes are all over the place. And so that's going to affect my normal microbiota. Now, during this whole COVID thing where we've done online teaching for months now, I'm sure my normal microbiota composition has changed because of that, because I'm no longer in a school environment, I'm no longer around hundreds and hundreds of kids. And so I'm sure that has had an effect on the normal microbiota that at least are living on me, and I'm sure within me as well, because I'm not breathing in all these different microbes that I normally would have if school had continued to be in session. So just an example of how your work environment can affect your normal microbiota. and the seasons of the year and so on. Are you going outside? Are you staying inside? Those are all things that can affect normal microbiota growth. Mentioned this a little bit ago, microbial antagonism. That's competition between microbes, and through that, they offer us protection by occupying niches that pathogens might otherwise occupy. So they have claimed you as their turf, and they say, sorry, this is our turf, not yours, go away. And so the pathogens never get the opportunity to colonize. They produce acids to protect their turf as well. So it's kind of like, they have some weaponry here to protect what they want for themselves. So it's not like they just land on you and just, oh, this is pretty nice here, and if somebody else comes in, I'll just move. No, they will produce these compounds, proteins, acids that that inhibit the growth of other bacteria, called bacteriocins. And so as a result of them producing these compounds, pathogens then cannot occupy that region of the body, whether it's in us or on us. So you should feel loved. You have microbiota that find you to be prime real estate, living on you and in you. And so if you ever feel sad or lonely or dejected, we haven't been in school for a long time and you might feel a bit lonely thinking, where is everybody? I don't have anybody. Remember your normal microbiota. They love you and they are competing for you. I'm being silly, but that's what's going on here. Your skin, your large intestine, it's all prime growing environments for these bacteria, and they will defend it by producing compounds that will reduce the chances of other microbes moving in and colonizing. So hopefully that makes you feel good about yourself. Probiotics are live microbes applied to or ingested into the body. They're intended to exert a beneficial effect. So look at the word probiotics. Pro in this case means for, it's in favor of, and then biotics, in favor of life. So these are things that are you can take compounds or whatever, or usually it's just it's microbes that you ingest. Like for example in this one, certain lactic acid bacteria can help alleviate diarrhea and prevent colonization by salmonella. Salmonella bacteria are very, very sensitive to pH. And so consuming these lactic acid bacteria sometimes helps in reducing salmonella numbers, prevents them from multiplying and dividing, and so your infection doesn't worsen. So there's a whole lot of probiotics that are out there. This term is like a catch term for just a number of foods in the food industry. Take probiotics, it's good for you. Take probiotics, it's good for you. And they can be. I mean, it's not like it's the answer to all of your illnesses, but probiotics can be helpful. You eat yogurt, I mean, that's a probiotic. You got all kinds of bacteria, live cultures that are living in yogurt. We ingest those. A number of them die, and some of them just don't do anything other than make yogurt what it is. But are they beneficial? Are they probiotic? Yeah, in some cases they say yes. Our tests and research have shown that they are definitely beneficial to our digestive system. You need to watch those. Take it with a grain of salt. It's not harming you. Is it helping? Are they helping us? It's hard to say. There's so much competition that's going on in our digestive tract that when you ingest these bacteria, their numbers are, I mean, you're taking, you would literally be eating millions of them, but compared to the billions upon billions that are inside our large intestine, I don't know if they stand a chance actually being able to colonize our gut and exert their beneficial effect because of the normal microbiota that have already colonized our digestive tract. So I'm a little skeptical about it, but it's not going to hurt you, of course. Just something you need to look into and be aware of. So there was some question out there. Okay, so we have all this normal microbiota. Is it necessary? Do we have to have them? So they did research with germ-free mammals. So they have no microbiota whatsoever. And researchers found that the microbes, the normal microbiota, are not essential for life. They have weak immune systems and require more calories and vitamins. They're weak immune systems obviously because they're in a sterile environment, and so they're not being exposed to anything, so the immune system is not being educated to fight off anything. And they require more calories and vitamins that are assisting in, if we have normal microbiota, they're assisting with that whole digestive process. because it's a mutualistic relationship, and so they require some additional supplements. However, they generally live twice as long as their normal counterparts, and obviously because they're not exposed to pathogenic microbes, so they're going to live longer. But anyway, it was just a kind of an academic question. Are microbes essential for life? No, we don't have to have them. They're not necessary for us to live. However, they definitely are exerting an influence over us. So there was this bacteriologist. He was a giant, not like in size. I mean, I don't know if he was a big guy or not. I think he looked from pictures, he looks like he's a normal sized guy. He was a giant in terms of his discoveries. Robert Koch, he was a German bacteriologist. And what was awesome was that he and Louis Pasteur were contemporaries. They were living together at the same time. So we had two of the most brilliant minds in bacteriology and virology and the germ theory of disease. They were both living simultaneously. They were competitive. They had definitely had a high level of respect for each other, but they were competitive in their research and who was going to discover this and who was going to discover that first. But both living in Europe, Robert living in Germany and Louis living in France. But what an awesome time for microbiology. And from them, I mean, we got the whole idea that there's no such thing as spontaneous generation, the germ theory of disease, these postulates here called Koch's postulates, which were used to determine the etiology or the cause of a disease. He figured it all out. And so through his research, we have what are called these Koch's postulates. It's 4 simple steps. The same pathogen must be present in every case of the disease. So this is a method. I'm sorry, I should back up and state. This is a method, again, to determine what is the causative agent of a disease and how do you prove it. And this is what does it. So in every case of the disease, the same pathogen must be there. It must be isolated from the diseased host and then grown in pure culture. So you isolate it, take it, grow it separately. The pathogen from the pure culture must cause the same disease when inoculated into a healthy host. So you take it out of pure culture, put it into another host. The pathogen must be isolated from the inoculated animal and be same as the original. Okay, so those are the 4 steps. Pretty simple, but let's make it a little more visual. So you take the dead animal and you isolate the microbes from it. So colonize the pathogens and also look at them and identify them under the microscope. Then take those pathogens and inject it into a healthy animal. Poor mouse, he's going to be curtains for him pretty soon. And like I said there in the statement, they're used to determine the etiology or the cause of infectious diseases. Then that animal dies, the second animal dies. You isolate the pathogens from that animal, identify them, and they need to be the same. It seems very logical and straightforward, but this was in the 1800s. This was at a time when people were not being logical and straightforward about disease. The prevailing idea about how we got infected and how we acquired a disease was the miasma theory. We just breathed in bad air, and that bad air made us sick. And it made us sick in a variety of ways, and you couldn't figure out how it did that. You just breathed in the wrong air, and it exhibited itself in some kind of an infection, which was ridiculous. And that was the prevailing thought for centuries, was the miasma theory. Well, along comes Robert Koch, and he says, wait a minute, there are bacteria that are causing disease. Particular bacteria will cause a particular disease. And people were like, what are you talking about? This is not the way it is. This is crazy. And so then he had to fight and prove and experiment to get his idea accepted. And this is what he did. So he and Louis both, and others, but those are the 2 big names then, he and Louis Pasteur both worked on defeating this whole miasma theory idea, which was garbage. And it took a lot of work, a lot of dedication in order to bury miasma and bring in the germ theory of disease, and that's what this was all about. Reproducing diseases in organisms and identifying these particular pathogens and saying this pathogen causes this disease and this pathogen causes this disease. And so this is what has led to the world that we live in today. The measles virus causes measles. Chickenpox virus causes chickenpox. That all came from Koch and the work that he did here. So it was a phenomenal foundational discovery that he made. So some terminology. There's quite a few, for quite a few terms, sorry. Symptom is a change in the body function that is felt by a patient as a result of a disease. This is subjective. So for example, whenever you go to the hospital and you see your levels of pain, that's a symptom. So very painful for one person could be just moderately painful for somebody else. So that's a symptom. It's a subjective thing. A sign is a change in the body that can be measured or observed. So this is quantitative. This would be like a fever. You can take somebody's temperature, get a number, it's quantified. So a fever is a sign. So that's the difference there. That's objective. You can see it, you can measure it, you can observe it. A syndrome is a specific group of signs and symptoms that accompany a disease. And typically we don't use this term syndrome for microbiological diseases, but occasionally it is. Typically the word syndrome, you may be thinking this, especially if you're in bio, is attributed to genetic diseases. Some kind of chromosomal mutation or a gene mutation or something like that. But it is occasionally used for microbiological diseases as well. A communicable and contagious disease. What's the difference between them? They seem similar. Communicable, a disease that is spread from one host to another. So it can be spread into the community, like herpes or typhoid or tuberculosis. But it's not as spread as easily as a contagious disease. A disease that is easily spread from one host to another, just through air droplets, for example. Chickenpox, measles, flu, COVID-19, those are contagious diseases, easily spread through airborne droplets that we release when we talk, speak, laugh, sing, whatever. Those are contagious diseases. Non-communicable diseases, a disease that is not transmitted from one host to another. Like, for example, tetanus, it gets into you through a puncture wound. You have to have something puncture your skin that has Clostridium tetani bacteria on it. And, of course, the classic rusty nail. Well, of course, it doesn't have to be rusty. Rust is not that doesn't lead to the disease. It's the bacteria that are on the nail or the thorn or whatever it happens to be. If it can puncture your skin and can seal up around it, that's the transmission of tetanus. So you don't get it from somebody. It's not like somebody can give you tetanus. They can't cough it onto you. They can't spit it onto you or anything like that. It's a non-communicable disease. So those are the differences between those three terms. Okay, that's the end of part 1. I'm going to stop there, and I'll be doing parts 2 and 3, so stay tuned for those, and thank you for watching.